Okay, thanks, Elson. And I think, yeah, again, my name is Sulabh Podial. I work with Ag Research, which is one of the public research organization based in New Zealand. And I'm going to share some of the progresses in terms of research, through our from our MFAT funded project that Tanya just kind of talked about, and some of the future news that we have identified. So a quick summary of regarding the history of CRB in the Pacific. I think by now, I think most of us know, that CRB, coconut rhinoceros beetle, was first found from Samoa in the Pacific back in 1909, and which is believed to have been introduced through rubber planting, transportation. So, and if we look at the like history of CRB invasion in the Pacific, there are three major time points. The first one, as I mentioned earlier, 1909, when it was first introduced, and then during 1960s, when a German scientist Fugger kind of first found out Oryctis nudibranchs, which infected CRB and was used as one of the major like management tools to manage CRB at that time. And then more recently, in 2007, from where like a new waves of invasion started in the Pacific, and this new like series of invasion was recognized to be associated with like previously unrecognized CRB, which was then called as CRBZ clade 1A. And interestingly, like all these populations in these newly invaded areas did show some degree of resistance, against the commonly used isolates of Oryctis nudibranchs, in short ORNV. Since 2007, there are, I think, more than 25 islands or, group of islands which has been, invaded by CRB in the Pacific, and the damage has been in the range of around like 40 to 90% palm damage in the newly invaded areas, with serious, impact on food security and livelihood. As you can see here, like I'm not going to like read all of them, but just to give you an idea about the impact of CRB on the livelihood and food security, as you can see, from Vanuatu to Hawaii, it has been really, devastating coconut palms all around. Okay, so in terms of research progress, some of the major progress has been around, identification of genetic markers to characterize the CRB population, which has been really helpful, not only to map CRB variants, but also to identify new invasive population, because in that way we are able to kind of optimize the management plans based on specific variants or specific like local CRB population. Similarly, we are also now able to identify biological control agents like genetically, and this has helped us to understand the background population of biological control agent in the wild, and whether the agent, whether the biological agent that we are releasing in the field are actually effective. In the meantime, Ag Research currently holds a solid database of more than 6,000 CRB samples from both native as well as invasive range, and which we have found, which is really being used extensively by both of our partners, and we do as well, and which we believe would serve as a good reference for any future studies. Next, a large part of our current work has been around screening and identifying isolates of both Oryctis nudibranchs and Metrasium against various CRB population. And currently, we have a good collection of biological materials again from both native and invasive ranges. And so far in this program, we have screened more than 10 isolates of Oryctis nudibranchs and around 7 isolates of Metrasium fungus in different countries, including Papua New Guinea, Solomon, Vanuatu, Guam, as well as Samoa. Based on both the lab studies as well as field results, we have identified at least 3 good Oryctis nudibranchs candidate. 2 of them were originally sourced from Philippines, whereas the third one was found from Madang in Papua New Guinea. Similarly, a couple of isolates of metrazium has also been has produced some really promising results in both Solomon and PNG, and are currently in the field release stage. A third on like surveillance system, there has also been some progress around use of publicly available in images to identify new incursions. There was like one of the paper that we published last year where we suspected a possible incursion of CRB in Timor-Leste and Mexico. Actually, with the help of some of the colleagues who are actually in the call right now, like Evel, we are now able to like confirm CRB specimen from Timor-Leste, whereas we are still working with our Mexican partners to confirm this further. So future needs, as you can see, like in the table in the right, that there is still 6 to 7 Pacific Island countries and territories which are free of CRB so far, and there are several picks where CRB is still restricted to few islands or island groups. So I think one of the urgent need is to develop a best practice response plans, because we have learned quite much, actually it has been quite a learning curve over the last decade or so. So we have learned so much, so including learnings from and experience from some of those recent invasion sites, and I think we have to develop a proper system for early identification and reporting. It is important because usually, as we have seen in the past, it takes time to get things together on after an incursion has been reported, and and most of the times we are usually late. So I think this could be like one of the major future needs. In terms of biological control agent, our research so far strongly suggests that we should find a proper pathogen to match with the local host for best results. For example, Port Moresby population, CRB population, has a different genetic background to those of Solomon Island and Vanuatu. And interestingly, this different population has been responding differently to the biological control agents. For example, we have found infection using a strain called V23B against CRB population from Vanuatu and Solomon in both lab and field releases, but the same strain was not effective against the Port Moresby population. So yeah, there is like this kind of like variation. Similarly, recently we have found a fungus belonging to the Beauveria genus infecting coconut rhinoceros beetle in Vanuatu. This is important, because this is the first time ever that we have found like any CRB samples infected by Beauveria, at least in our collection. Therefore, I think this research, these research are needed to find an appropriate biological control agent specific to a local outbreak. Because until we know those answers, the management approach that we take might not be that effective as we wish. Regarding the genetics of CRB and the pathogen, actually, Alison did kind of, put a note when she started. There's going to be a workshop in July in Solomon Islands. So yeah, so that's something that like to look forward for much more detail in that topic. Further, more on the like future research, one thing we still need like is kind of improved tools for surveillance and control. This could include a much more robust remote surveillance methods, tools to locate beetle population effectively. Example, it could be like some kind of biosensors. Next to pheromones, while CRB pheromone have been good enough so far for monitoring and detection of new invasion, it is still not that effective, for mass trapping as we see for others like, fruit fly. So I think there is a need to make it, much more effective and attractive. And next, sanitation. Again, we all know, that sanitation is one of the most effective ways to control and manage CRB. But again, doing manually is too labor intensive, and in many cases, almost impossible, because especially if there are too many dead palms already. So I think we need to look for some innovative tools or ways to do sanitation, and some of them, options could be, testing some degrading fungus, to degrade, the dead palms, or maybe using some kind of repellents so that the beetles are not attracted towards the dead palms, or some kind of fancy incinerators or crushing machines. So yeah. I think another area of research that is currently very thin is our understanding of the population variation in the native region of South and Southeast Asia. I think there has been some work done, but again, very few and far between. So it's something like that is needed. Also, I think looking at the biodiscovery approach, to look into other potential biocontrol candidates in the region, because I think for sure, there are others that have much more potential to manage CRB. With that, I would like to acknowledge the whole team of Ag Research team and beyond, and all of our collaborators from Pacific as well as South and Southeast Asia. With that, thank you, and hopefully I'm on time, Elson.